Good morning, students. In the today's lecture, we are going to discuss about the life cycle of the mucor. Okay, in the last lecture, we have discussed the life cycle of the albugo, and today's topic for the discussion is the life cycle of the mucor. Okay, the first of all, we are going to discuss about the systematic position or the classification of the mucor. Here, we are following the classification which is proposed by the Alexopoulos and the memes. As per the classification proposed by the Alexopoulos and the meme, the mucor it is placed in the kingdom Mycete. Okay, we have already discussed that. As per the classification of the Alexopoulos and the memes, all the fungi these are included in the kingdom Mycete. Okay, here the classification of the mucor it is included in the kingdom Mycete. Division A Mastigomycota, subdivision Zygomycotina, class Zygomycetes, order Mucorales, family Mucoraceae, and the genus is the Mucor. Okay, the classification of the Mucor here, the kingdom Mycete, division Amastigomycota, subdivision Zygomycotina, class Zygomycetes, order Mucorales, family Mucoraceae, and the genus is the Mucor. Okay, then the regarding the habit and habitat of the mucor. Okay, here the habit and the habitat of the mucor. The genus mucor, the term mucor or the name of that fungi mucor, it is derived from the Latin word museo. Okay, it is derived from the Latin word museo, and the meaning of the that Latin word museo, it is the bee moldy. Okay, the genus mucor it is represented by about more than 100 species, and these species these are reported to be found throughout all the world, but India only the 70 species of the mucor these are reported. Okay, the genus Mucor it is the most common member of the family Mucoraceae, and about more than 100 species of the mucor these are reported throughout the world, and of these 100 species only 17 species of the mucor these are reported from India. The mucor it is popularly and commonly called as the pin mold or the black mold. Okay, pin mold or the black mold. Okay, then the most of the species of the mucor that is these are saprophytic, these are present on the variety of the materials including the foodstuffs. Okay, in the damp conditions they generally spoil the organic materials such as bread, jams, jellies, and also the overripe fruits. Okay, the most of the species of the mucor are saprophytic, and these are growing on the variety of the material including the foodstuffs. In damp conditions they spoil the organic materials such as the bread, jams, jellies, and overripe fruits, also the syrups. Some of the species of the mucor that is, these are the coprophilous. These are coprophilous means that is these are dung inhabiting, and very few species that is these are strictly terrestrial, while there are certain species, for example mucor mucido and mucor racemosus, that is these are the common air contaminants. Okay, mucor mucido and the mucor racemosus, these are the common air contaminants. The mucor it can be grown in the laboratory by placing the bread slices in the moist chamber at the room temperature. Simply we can grow the mucor by putting a slice of bread in a moist chamber at the room temperature. Okay, and after 2 to 3 days if you observe that container, then you will see that is there is a formation of a white cottony or the woolly growth on that particular slice of the bread. That white cottony or woolly growth it is nothing but the mucor. Okay, that is how the mucor it can be grown in the laboratory, even in the home. Just take a slice of a bread, and that slice of a bread the kept in a moist chamber. and then after 2 to 3 days you will observe that there is a formation of a white cottony or the woolly growth on that slice of the bread. That white cottony or woolly growth, it is nothing but the mucor. Okay, then the species of the mucor, these are known to the cause. There are certain species of the mucor which are known to cause a disease which is called as the mucoromycosis in the domestic, also domestic animals, and also it is known to cause the mucoromycosis in the human being. Okay, there are certain species of the mucor, these are known to cause mucoromycosis in the domestic animals and the human being. Okay, in this disease, the mucor, it will infect the vital organs. The mucor will infect the vital organs like the lungs, then brain, etc. Okay, and ultimately it may cause the death of that organism. Okay, in the mucormycosis, the mucor, it will infect the vital organs like the lungs and the brains, and even in the severe cases it will be responsible for the death of the organism. Okay, that is about the habit and the habitat of the mucor. Okay, then here you can see the vegetative structure of the mucor. The thallus of the mucor, it consists of highly branched cenocytic mycelium. Okay, here you can see the vegetative mycelium of the mucor, and it is highly branched structure. Here you can see that is the vegetative mycelium of the mucor. It is highly branched and it is cenocytic. Why it is said to be the cenocytic? Because it is aseptate, that is there is a complete absence of the partition walls in the hyphae, and here the large number of the nuclei, that is these are scattered in the hyphae without any partition wall, and that's why the vegetative mycelium of the mucor, it is said to be the cenocytic. Okay, and that mycelium, it is forming the white or gray or the brownish colored cottony growth on the substratum. Okay, the mycelium, it is forming the white, gray or brownish cottony growth on the substratum. Here in this one, here you can see that is the mycelium, it is differentiated into the two mycelium, it is differentiated into the aerial hyphae. Okay, here you can see that is the aerial branches, and here these are the subterranean hyphae, or we can say that is these are the prostrate hyphae, and these are the aerial hyphae. Okay, the mycelium of the mucor, it is differentiated into the aerial and the prostrate hyphae. Okay, aerial hyphae, and it is the prostrate hyphae. Okay, then the prostate hyphae. Okay, here you can see the function of the prostate hyphae, that is it serves the function of absorption. Okay, the absorption of the food material from the substratum on which, that is this mycelium, it is growing. Okay, that is the function of the prostate hyphae, that is the absorption of the food material from the substratum on which the mycelium of the mucor, it is growing. Okay, the young hyphae, that is these are without any septa, that is these are not having any the partition wall, but when they become older, then there is a formation of the septa or the partition walls in the hyphae. Okay, when they mature, then the septa or the partition walls can be observed in the hyphae. Okay, then the if there is a anaerobic conditions, that is for example in the anaerobic liquid cultures, that is especially when there is a presence of the abundant carbon dioxide, the hyphae, it will break down into the yeast like bodies. Okay, whenever in the anaerobic liquid medium, when there is a abundant amount of carbon dioxide available, then at that time the hyphae, it will break down into the small yeast like cells. Okay, yeast, that is also called as the saccharomyces, that is the unicellular fungi. Okay, in the anaerobic condition, when the carbon dioxide is abundant, then at that time the mucor, the hyphae of the mucor, it will appear as a yeast like cell. Okay, and this state of the fungus, it is referred as the torula condition. Okay, torula. This is like condition in the mucor, it is called as the torula condition. And if the oxygen it is available in the sufficient amount, then the it will reverse it shape as per the it will become filamentous. Okay, it will reverse to the filamentous state. That condition we called it as a torula condition, torula condition, torula condition. Then another important thing here, the cell wall, that is the cell wall of the hyphae, it is made up of chitin. Okay, it is made up of chitin and chitosan. Okay, and beside these chitin and the chitosan, there is a deposition of the polysaccharides, then proteins, lipids, purines, pyrimidines, and also the few minerals in the cell wall. Okay, principally the cell wall of the hyphae in the mucor, it is made up of the chitin and the chitosans, but along with this there is a deposition of the polysaccharides, proteins, lipids, purines, pyrimidines, and a few minerals in the cell wall. Okay, that is about the vegetative structure of the mycelium in the mucor. Okay, then the reproduction in the mycelium, it takes place by the three methods. The first one is the vegetative reproduction, the second one is the asexual reproduction, and the third one it is the sexual reproduction. Okay, we are discussing here the reproduction in the mucor. The reproduction in the mucor, it takes place by the three methods. The first one is the vegetative reproduction, second one is the asexual reproduction, and the third one it is the sexual reproduction. Vegetative reproduction here in the mucor, it takes place by the fragmentation. Okay, vegetative reproduction in the mucor, it takes place by the fragmentation. Due to the accidental breakage, the mycelium it may break up into the two or more units. Okay, due to the accidental breakage, the mycelium it may break into the two or more units, and each unit is capable to grow as the mother mycelium. Okay, that is how the vegetative reproduction by the fragmentation it occurs in the mucor. Okay, due to the certain accident, due to the mechanical injury, it get breakdown into the two or more units, two or more fragments, and each of the fragment has the capacity to divide, redivide, and to develop into the new mycelium of the mucor. Okay, that is about the vegetative reproduction in the mucor. Then the second method of reproduction, it is the asexual reproduction. Asexual reproduction in the mucor, it takes place by the formation of the sporangiosspores. Sporangiosspores, these are also referred as the aplanospores. Okay, and another method of asexual reproduction, that is there is a formation of the another type of the spores which are referred as the chlamydospores. Okay, two types of the spores are developed in the mucor during the asexual reproduction. That is the first one, these are the sporangiospores which are also referred as the aplanospores, and another type of the spores, that is these are the chlamydospores. Okay, the first of all, here in this photograph you can see we are going to discuss about the asexual reproduction by the formation of the sporangiospore or the aplanospores. Okay, here you can see that is this is the mycelium of the mucor, this is the prostrate hyphae, and these are the erect hyphae, or these are referred as the aerial hyphae. Okay, here the asexual reproduction by the formation of the sporangiosspores or the aplanospores, it is the most common method of the multiplication in the mucor. Sporangiospore formation or the aplanospore formation in the mucor, it is the most common method of the asexual reproduction in the mucor. Okay, here the hyaline branches of the mycelium which bears the sporangia at their tips, these are known as the sporangiophores. Okay, here you can see that is these are the aerial branches, and at the tip region of the aerial branches there is a presence of the sporangia, and that's why these aerial branches bearing the sporangia, these are referred as the sporangiophores. Okay, in some of the species the sporangiophores are branched. Here you can see these sporangiophores are unbranched, but in some of the species these sporangiophores may be branched. Okay, in such cases, the terminal sporangium is larger than the laterals. Okay, then the tip of the sporangiophores. Here you can see that is how exactly the sporangia these are developed. Here you can see that is the tip of the sporangiophore swells. Here you can see that is the swollen hypal tip. That is the aerial hyphae, and the tip of that aerial hyphae it will get swollen because where it accumulates the large amount of cytoplasm along with the large number of the nuclei, and that's why the tip of the aerial hyphae it get swollen. Okay, and then the swollen tip it will gradually enlarges and it will develop into the sporangium. Okay, that tip region, here you can see this is the tip region, it will gradually enlarges in the size and later on it will develop into the sporangium. Okay, it will develop into the sporangium. Okay, towards the maturity the content of the sporangium, here you can see towards the maturity, that is it enlarges in the size, and towards the maturity the content of that tip region it will get differentiated into the distal multinucleate densely cytoplasmic spore plasm. Okay, here you can see that is this region, it is get differentiated into the outer region. Okay, that is the outer region, it is having the abundant cytoplasm or it is filled with the dense cytoplasm, it is having the large number of the nuclei, and that particular region it is called as the spore plasm. Okay, it is said to be the spore plasm. Okay, and the cylindrical portion, it is the central cylindrical portion, it is a, sterile portion because it is not having the any type of the nuclei. Okay, because all the nuclei, that is these are migrated into this spore plasm, and this central cylindrical portion it is devoid of the nuclei, and that portion it is called as the columella plasm. Okay, it is called as the columella plasm, and ultimately it will develop into the dome shaped structure, and that dome shaped structure it is referred as the columella. Okay, that means the mature sporangium it is differentiated into the spore plasm and the columella plasm. From spore plasm sporangia is developed, and from columella plasm columella it is developed. Spore plasm, because in this region the spores are developed, it is the fertile region, but here the columella region it is a divide of any nuclei, and that's why it is the sterile region. Okay, it is sterile region. Okay, then the content of this spore plasm, the content of this spore plasm it is cleared into the large number of 2 to 3 nucleate spores. Okay, generally the spores, this spore plasm it will develop into the spores. Here you can see the spore formation takes place. Here in this one you can see that is the spore formation takes place, but generally the spores in most of the species of the mucor, that is these are 2 to 3 nucleate. Okay, 2 to 3 nucleate. All these spores are almost equal in the size. Okay, the spores which are developed in the sporangia, these are 2 to 3 nucleate, and all these are morphologically similar, or we can say that is these are almost equal in the size. Then what happens, how the dehiscence of this sporangium takes place and the spores are dispersed into the outer environment, that is at maturity. At maturity means when the spores are developed in the sporangium, what happens, the sporangial wall here it get completely dissolves, and the spores, that is these are usually attached to the columella. Okay, though the sporangial wall it is dissolved, the spores, that is these are attached firmly to the columella. and these are dispersed very slowly by the wind or by the ants or the insects. Okay, the sporangial wall dissolves, but here the spores, that is, these are firmly attached to the columella, and these are slowly dispersed by the wind, ants, or the insects. Okay, that is how, that is, these are liberated into the outer environment. Here the spores, that is, these are dark, these are dark colored, these are usually elliptic to ovoid, and these spores germinate when they fall on the suitable substratum by the germ tube and form the new mycelium. Okay, whenever these spores, these are dispersed into the outer environment, whenever the conditions are favorable, they will germinate, they will form the germ tube, and later on they will develop into the new mycelium of the mucor. Okay, that is how the asexual reproduction, it takes place by the formation of the sporangiospores or the aplanospores in the mucor. Okay, then the asexual reproduction by the formation of the chlamydospores. During the unfavorable conditions, the thick-walled, nutrition-rich, intercalary mycelium segments, these are developed by the septation of mycelium, and these are termed as the chlamydospores. Okay, generally during the unfavorable conditions, that is, the large amount of food material, it get accumulated into the intercalary mycelium, it will secrete the thick wall, and that particular structure, it will be referred as the chlamydospores. These chlamydospores, they will get separated from each other when the connecting mycelium dries up. Due to the unfavorable conditions, the connecting mycelium, it will dry, and ultimately the chlamydospores, these get separated from each other. And whenever the conditions are favorable, that means the temperature, it is optimum, whenever the sufficient amount of moisture is available, then at that time the chlamydospores will germinate and give rise to the new mycelium. Okay, remember that the chlamydospore formation, it takes place during the unfavorable conditions. Unfavorable condition, that means the proper temperature is not there, then the amount of moisture, optimum moisture is not available, then at that time what happens, the intercalary mycelium, it will accumulate a huge amount of food material, then it will secrete the thick wall around itself, and then it will live in the unfavorable environmental conditions, and that particular structure, it is called as the chlamydospores. Okay, but due to the unfavorable conditions, what happens, the connecting mycelium will rise up, and ultimately the chlamydospores are separated from each other. But when the conditions are favorable, that is, optimum temperature is there, then the sufficient amount of moisture is available, then at that time these chlamydospores will germinate, and then they will give rise to the new mycelium. Okay, that is all about the asexual reproduction by the formation of the chlamydospores. Then the third method of the reproduction in the mucor, it is the sexual reproduction. Okay, the sexual reproduction in the mucor, it is a isogamous type. Isogamous type means, here you can see that is, these are the gametangia, and these are morphologically similar. That is, these are identical in their shape, size, and the color. That's why we cannot differentiate them as the male gamete and the female gamete. It is considered as the primitive type of the sexual reproduction. Okay, the sexual reproduction in the mucor, it is isogamous. That is, the copulating gametangia, these are morphologically similar. Okay, the species of the mucor, these are either homothallic or these are heterothallic. Okay, the species of the mucor, these are either homothallic or heterothallic. In a homothallic species, the zygospores, these are developed by the mycelium, which is derived from the single sporangiospore. Okay, just we have discussed the sporangiospore, that is, whenever the mycelium, these are developed from the single sporangiospore, and the in this mycelium, when the sexual reproduction takes place, it will be called as the homothallic species. Okay, but Most of the species, these are heterothallic. Okay, in such species, the zygospores are formed only when the mycelia of the two strains meet. Okay, here you can see that most of the species of the mucor, here the mycelia of the two strains, that is they get fused with each other, that is these are the positive strain and the negative strain. That means these are developed from the two, the mycelia, these are developed from the two different sporangiospores. Okay, here that is these hyphae, that is these are described as the positive and the negative strain. Okay, in this one here you can see that is this is the positive strain and the negative strain. Okay, the hyphae of the different strains, that is these are morphologically similar, but these are physiologically distinct, these are physiologically different. So here you can see that is these are morphologically identical, but these are physiologically distinct. The sexual process in the mucor, it involves the three major steps. Three major steps in which the sexual reproduction in the mucor occurs. Here the first one, the initiation of the progametangia. Okay, here you can see the initiation of the progametangia. Then growth of these gametangia towards each other. Okay, that means the first of all, first step, initiation of the gametangia. Second one, growth of gametangia towards each other. And the third step, that is the fusion of the gametangia. Okay, fusion of the gametangia to form the zygospore. Okay, the three steps in the sexual reproduction of the mucor. The first one, it is the initiation of the gametangia. Second one, the growth of gametangia towards each other. And the last one, the fusion of the gametangia to form the zygospore. Okay, then there is a special hormone which is present in the mucor, which plays an important role in the process of the sexual reproduction, and that hormone, it is the trisporic acid. Okay, trisporic acid is the hormone which is present in the mucor, which plays an important role in the sexual reproduction of the mucor. Okay, remember it, trisporic acid. Okay, that is all these processes, that is here we are discussing the sexual reproduction in the heterothallic species. Okay, the heterothallic species. Okay, here the sexual process, it will begin with the growth of the hypal branches of the positive and the negative strands towards the each other. Okay, here you can see that is the growth of positive strain hyphae and the growth of the negative strain hyphae towards the each other. Okay, and that is how the sexual process it is initiated. The tip of these hyphal branches come close to each other and they develop into the progametangia. Okay, the tip of these hyphae, that is they will touch with each other, and thus that is these structures, that is these are called as the progametangia. The large number of the nuclei along with the cytoplasm, it will move into the progametangia. Okay, from the hyphae, the large number of the nuclei and the abundant amount of the cytoplasm, it will move into the progametangia. Okay, then there is a formation of a transverse septum into each progametangium. Okay, here you can see there is a formation of a transverse septum to the near the tip region in both the progametangia, in the both gametangia. Okay, the tip region, here you can see that is it is the basal region, it is the proximal part, that is called as the suspensor. Okay, it is the basal part of the progametangia and it will form the suspensor. Okay, since the gametangia possesses the undifferentiated mass of the multinucleate protoplast, that is these gametangia, these are now called as the cenogametangia. Okay, that particular tip region, it is having the abundant amount of the nuclei and it is undifferentiated mass, and that's why now these gametangia, this tip part region, it is said to be the cenogametangia. Okay, these are called as the cenogametangia. The walls between the two conjugating gametangia dissolves. here you can see that is the wall, it is the suspensor, and it is the mature gametangia. And when they get matured, then at that time the wall in between these gametangia, it will get dissolves, and ultimately the protoplast of these two gametangia get fused with each other, and that the formation of the fusion cell, it is called as the xenozygote, or it is called as the zygospore. Okay, the walls between the two conjugating gametangia dissolves and the protoplast mix, resulting in the formation of the fusion cell, and this called as the zygospore. In some of the book you can find that is this zygospore, it is also referred as the cenozygote, COENOZYGOTE. Okay, zygospore, it is referred as the cenozygote. As both the conjugating gametangia, these are identical in the morphology. Hence, the process of conjugation in this, it is known as the isogametangial copulation. Okay, and here the both these gametangia, these are morphologically similar, that means these are identical in their shape, size, and the color, but these are physiologically different, that is these are, one is the positive strain and the another one is the negative strain. That's why here this process, this sexual reproduction, it is said to be the isogametangial copulation. Okay, because here the entire content of these gametangia, it get mixed with each other, and that process, it is said to be the isogametangial copulation. Okay, then in the zygospore, the nuclei of the opposite strands fuse in pair. Okay, in the zygospore you can see that there is a presence of the nuclei, but these nuclei of the opposite strand, that means some of the nuclei, that is these are positive strand, and the other nuclei, these are of the negative strain. Okay, then they will fuse in the pair, that means the nuclei of the opposite strand, they will get fused with the each other after the plasmogamy, and these are forming the large number of the nuclei inside the zygospore. Okay, the nuclei of the opposite strain in the zygospore will get fused with each other, and there will be the formation of the large number of the diploid nuclei in the zygospore. Okay, meanwhile the size of the zygospore, it will enlarges, and it will secretes a thick wall. Okay, it will secrete a thick wall, that is it is differentiated into the exospore and the endospore. Okay, meanwhile the size of the zygospore, it will enlarges, and it will secrete the thick wall. The outer one, it is called as the exospore, and the inner layer of that thick wall, it is called as the endospore. Okay, the endospore, it is made up of three thin elastic layers. Endospore, it is again, it is made up of three thin elastic layers, whereas the exospore, it consists of the two brown or the black layers. The zygospore, it remains along the substratum during its the development. Okay, then what happens, all the diploid nuclei which are formed in the zygospore after the plasmogamy undergo the meiosis division. Okay, that is the third stage in the sexual reproduction. The first step here, the plasmogamy takes place, that is the entire content of the gametangia get mixed with each other. Then in the zygospore, the second step, it is the karyogamy, that is the nuclei of the opposite strain get fused with each other, and there is a formation of the large number of the diploid nuclei. And the third stage in the sexual reproduction, that is the meiosis. Okay, all these diploid nuclei formed in the zygospore after the plasmogamy, that is they will undergo the meiosis. The zygospore, it usually undergoes the long period of the rest, and thus the resting zygospore has the many haploid nuclei. Okay, the zygospore, it will undergo the long resting period, and the resting zygospore, it is having the large number of the haploid nuclei. Then at the time of germination of the zygospore, the exospore, that is the outer wall of the zygospore, it will rupture, and the endospore, it grows out in the form of the long thin tube-like structure. Okay, what happens, at the time of germination, the exospore, it will get ruptured, and the endospore it will get developed into the long thin tube like structure, and it is called as the sporangiophore. Okay, it is called as the sporangiophore. That is, all the haploid nuclei which are present in the zygospore, they will migrate to the tip of the that sporangiophore, and consequently, due to the accumulation of the large number of the nuclei in the tip region, that tip region it will swell, and now that sporangium it is called as the zygosporangium. Okay, now it is called as the zygosporangium. Rarely more than one sporangiophore arises from the single zygospore. Okay, rarely more than one. Generally, only one sporangiophore it is arised from the zygospore, but rarely more than one sporangiophore. These are arised from the single zygospore. Okay, however, that is the sporangiophores. That is, these may be branched. Okay, and one also these may be unbranched. Okay, that is how the new mycelium of the mucor it is developed by the sexual reproduction. Okay, we have discussed in this lecture about the life cycle of the mucor. In this one we have discussed the systematic position or the classification of the mucor, which is proposed by the Alexopoulos and the Mimes in the 1979. Then we have discussed the habit and habitat. Then also we have discussed the vegetative structure. Then the reproduction it takes place by the three methods. The first one, vegetative reproduction by the fragmentation. Then second one, asexual reproduction by the formation of the sporangiophores and the chlamydospores. And the third one, that is the sexual reproduction, which is isogamous. Okay, now the here the diagrammatic representation of the life cycle of the mucor. Here you can see that is the vegetative mycelium of the mucor. Then here, that is how exactly the sporangiophores are developed. We have discussed this one. Okay, sporangiophores are developed, then on the tip region of the sporangiophores, that is the sporangia are developed. Okay, here the sporangia are developed, then here these spores, these sporangiospores, that is these are liberated into the outer environment. Okay, these are the sporangiophores. That is, these sporangiophores, whenever they get the favorable environmental condition, they will germinate, they will form the germ tube, and ultimately the new mycelium of the mucor it is formed. Okay, then the asexual reproduction it is also takes place by the formation of the chlamydospore. Here you can see that is the intercalary mycelium. It accumulates the abundant amount of food material, it secretes the thick wall, and that is that particular structure it is referred as the chlamydospores. These chlamydospores, that is, they get separated from each other. Then whenever the conditions are favorable, they will form the germ tube. Then after the germination, they will develop into the new mycelium of the mucor. Okay, that is the this one, this is the asexual reproduction by the formation of the sporangiospores, and this cycle it is representing the asexual reproduction by the formation of the chlamydospores. Okay, there it is, you can see the sexual cycle. Okay, here you can see the first of all vegetative mycelium, how exactly the gametangia are formed. Then these gametangia of the opposite strain get fused with each other. Okay, that is the isogametangial copulation takes place. That is the entire content of these isogametes get mixed with each other. That is the plasmogamy takes place. After the plasmogamy, karyogamy takes place. That is the nuclei of the opposite strain get fused with each other, and then the large number of the diploid nuclei are formed, and that is how the zygospore it is developed. Then here the in the diploid nuclei in the zygospore, they will undergo the meiosis division, and there will be the formation of the large number of the haploid nuclei. Okay, then the exospore, that it get ruptured, the in endospore it will form the thin tube like structure, that is called as the sporangiophores. Then the nuclei, they will move into the tip region, it will swell, it is called as the sporangiospores. And that is then the here, that is these spores, as these are formed due to the meiosis, that is these spores are referred as the meiospores. Okay, these spores, these are formed due to the meiotic division which takes place in the zygospores. These spores are called as the meiospores. These meiospores, then whenever they get the favorable environmental condition, they will germinate, they will develop the germ tube, and ultimately then they will develop into the new vegetative mycelium of the mucor. Okay, that is all about the life cycle of the mucor.